Thank you. Thanks to everyone for attending today, and thank you to the KAT6 Foundation for the invitation to speak. So my name is Jill Fahrner. I direct the Epigenetics and Chromatin Clinic at Johns Hopkins, and I'm a clinical geneticist and researcher. So we see patients with kind of the KAT6 syndromes, but also the broader group of these Mendelian disorders of the epigenetic machinery in our clinic. And then my lab works on a subset of these disorders in mouse and human cellular models. So today I'm actually going to be giving a very different talk than anyone else because I'm going to talk a little bit about KAT6 syndromes, but put them in the context of this larger group of Mendelian disorders of the epigenetic machinery, and then give some perspectives from our clinic, as well as some of our work on a different disorder within this group. So no disclosures. So my objectives for today are to define the Mendelian disorders of the epigenetic machinery, to describe how genetic variation can have epigenetic consequences, catching up here. To discuss the implications for treatment, and then to introduce our clinic. So, the Mendelian disorders of the epigenetic machinery are a group of genetic disorders that result from germline pathogenic variants in genes encoding components of the epigenetic machinery. So here I'm showing this large wheel, which shows the 85 genes that were known to cause these disorders back in 2023. And they're divided based on whether the genes encode enzymatic writers of epigenetic marks. Can you see that? Enzymatic erasers of these marks, enzymatic remodelers, or non-enzymatic readers. And try to use the pointer. Okay, there we go. Is that working? There we go. And some of these have actually more than one function. So some they can have an enzymatic writer function, a remodeler function, or an eraser function, but sometimes they also have a reader function, which is a non-enzymatic function, and those that do are shown in gray, and we refer to those as dual function components. And both KAT6A and KAT6B, which we've heard a lot about today, are both writers of a particular epigenetic mark, but they can also read particular epigenetic marks. So, as we've heard a lot and we all know, the clinical features of these disorders include intellectual disability as one of the most common features, and you can see those that have intellectual disability are highlighted in blue around the periphery. Growth abnormalities are kind of the second most common feature of the group in general, and those that have growth abnormalities are shown highlighted in orange around the periphery. And then in addition to these two most common features, we know that there are also many different tissue diverse manifestations in these disorders, including other neurobehavioral findings that we've heard a lot about today, as well as structural anomalies in various organs, eye findings, musculoskeletal features, and many other. So I don't know why it's not advancing. Yeah, I am. Oh no. Oh, there we go. Okay. So these disorders are autosomal dominant. We've heard a lot about that, and that's indicated here by the filled circles around the periphery. So many of these disorders are dominant conditions, meaning you only need a variant on one allele to have the disorder. And many of these occur on the autosomes or the non-sex chromosomes. And as we've talked about, KAT6A and 6B are writers and readers. And so now I want to get into a little bit more because we know that these are genetic disorders, but we've also heard some today about epigenetic consequences. And so I want to explain a little bit about how this group of disorders are actually genetic disorders, but they have these epigenomic consequences. So, as we all know, pathogenic variants in KAT6A cause Arboleta-Tam syndrome, and those in KAT6B can cause this spectrum of conditions ranging from genitopatellar syndrome to, say, Barber-Biesiger-Young-Simpson syndrome. And so, what happens, just to kind of broadly put it all together, is you have a variant in a particular gene. But that gene encodes a protein, and this protein is a writer and reader of epigenetic marks. And so when that's not functioning properly, we have the features that we see in these disorders. So as we've talked about, KAT6A and 6B are writers of epigenetic marks, and they switch genes on in cells by adding a chemical group called acetylation to the histones, and that's just being shown here. And you can see that when they add this group, this acetylation mark leads to a more open chromatin state and is associated with gene expression. And we know that there are these opposing writers and erasers of this mark, and when this mark is not present, this leads to a more closed chromatin state and gene silencing. So this is very important for gene expression. And when we have variants in these genes that disrupt their function, and we've talked a lot about what these variants do today, and it's still sort of unclear, and it may not be so simple as, all variants cause a disease by the same mechanism. We think there may be many mechanisms, and there may, in fact, even be, as Dr. Arboleta alluded to, a couple of disorders within this group. But to kind of simplify things, if a writer of a particular mark is not functioning properly, then you can have sort of too much eraser activity, and this sort of tips the balance of chromatin and can alter gene expression. But it's also not as simple as one mark on one gene. So these Mendelian disorders of the epigenetic machinery, even having a change in one of these epigenetic components can lead to many, many changes in gene expression and can have both direct and indirect effects. So I just want to illustrate this concept here. So in the normal setting, some genes like gene 1 are expressed and other genes like gene 2 are silenced. So genes like gene 1 their promoters are characterized by writers of activating marks, erasers of silencing marks, readers of these marks, and then these chromatin remodelers that actively alter chromatin structure. And then the net effect of this leads to an open chromatin state, which is shown here, and gene expression. Other genes like gene 2 are characterized by having writers of silencing marks, erasers of activating marks, and readers of these marks, with the net effect being a more closed chromatin state and gene silencing. But when we have a Mendelian disorder of the epigenetic machinery like KAT6A or KAT6B, we have a particular component that's not functioning properly. So, for example, a writer that's not functioning properly. And just to assume that this is a loss of function variant, if you don't have this writer functioning properly, it can't set up the proper chromatin state to lead to gene expression. And thus, there can be this sort of abnormal chromatin state and abnormal gene silencing of a gene that should be expressed. And this is the direct effect. But there can also be many indirect effects. And so what I mean by that is that if, for example, you have this abnormal gene silencing and this abnormal chromatin state, components of the epigenetic machinery that should be present at other genes like gene 2 might be drawn away, and there may not be sufficient amounts of them. And so then genes like gene 2 have a distinct but still abnormal chromatin state that's a more open chromatin state and leads to abnormal gene expression. And so this would be an indirect effect. And so you can see that there can be these direct and indirect effects, and that this can happen at many genes throughout the genome, and this can cause broad epigenomic changes and broad changes in gene expression, leading to the features of these disorders. So we've talked a lot today about the clinical features of these disorders, but I'm not going to go through that again, but I just want to emphasize that we think it's these changes in gene expression, kind of throughout the genome and in different cell types that can lead to these different features of the disorders. And so Now I want to kind of switch gears a little bit and talk about our epigenetics and chromatin clinic at Johns Hopkins, which is now multidisciplinary and where we see patients with this broader group of conditions. I'm the clinical geneticist and the director. Dr. Harris is the neurologist and neurodevelopmental specialist, and then our genetic counselor is Jackie Britton. And then when we need to do neuropsychological testing, Rowena Eng does that over at our institution at institution right across the street called Kennedy Krieger. And so we kind of compiled our clinical data, and in 5 years, we saw 432 patients, and over half of those were outside of the state of Maryland. And we saw individuals from 3 different continents, so North America, South America, and Australia. And you can see that people sort of have come from many different parts of the country to our clinic. We've seen when we compiled this data a few years ago, we had seen about 153 patients with epigenetic conditions, which include the Mendelian disorders of the epigenetic machinery shown here in red, as well as imprinting disorders and some other that don't fit into either group. And then when we actually looked at the top diagnoses that we saw, Arboleta-Tham syndrome was the kind of the 4th most common condition that we saw in our clinic. And 115 patients in this broader group of disorders had pathogenic variants in 26 distinct epigenetic machinery genes. And the vast majority had variants in writers of epigenetic marks, followed by erasers, readers, and remodelers. So we're seeing a wide variety of individuals in our clinic, and this kind of allows us to really see kind of similarities and differences between the different disorders. Oh, sorry, this one didn't translate very well from Mac to PC. But what Dr. Harris compiled the neurodevelopmental phenotypes of the different disorders that we saw in clinic. And one of the takeaways from this slide is that cognitive impairment was really present in 100% of individuals with Mendelian disorders of the epigenetic machinery. And this ranged from cognitive impairment that sort of didn't quite qualify as intellectual disability to severe to profound intellectual disability. And unfortunately, this is sort of completely distorted. So I'm going to just move on from this, but just to say that these Mendelian disorders of the epigenetic machinery have very significant cognitive effects on patients as well as behavioral, and so we're able to really see these similarities and differences between the group by seeing kind of this broad group of conditions. So we know that the current management of these disorders for KAT6A and 6B, of course, but also for the broader group is really just symptomatic treatment. So early, and Dr. Harris, I think, used this slide earlier. So early intervention, developmental therapies, and educational support, and certainly sign language and communication aids. And then we also do a variety of screening, looking for any other internal anomalies like echocardiograms, looking for cardiac disease, eye exams, symptomatic treatment of GI manifestations, sleep studies, if there's a concern for that, for any sleep abnormalities, abdominal ultrasounds, in some cases, brain MRIs and audiology evaluations. But the point is that we really don't have a treatment that gets at the underlying cause and that we have to treat all these symptoms individually. But what we hope is that someday we actually may be able to find a treatment for this. And so this is just a slide to kind of introduce kind of a treatment approach that is distinct, I think, from what we've heard today, and something that we've been working on in our lab. So just to kind of give the example of the KAT6 syndromes. So as we've talked about, the KAT6A and 6B places this acetylation mark. And if you don't have that, that can sort of tip the balance of chromatin in one direction. But it's not quite as simple as kind of just loss of this activating mark leading to more closed chromatin and gene silencing. We see these direct and indirect effects that I talked about previously. But one concept that we had come up with is we know there are some epigenetic small molecule therapies that exist out there that were initially developed to treat other diseases, but that in some studies had been shown that if you can sort of counteract the effect of the abnormal chromatin state from these disorders. So, for example, if you have a defect in acetylation, if you could then sort of alter the chromatin back in the other direction with a drug that sort of inhibits the opposite component of the epigenetic machinery. So you have a defective writer. If you could sort of inhibit the opposing eraser, you might be able to restore this balance of chromatin. And so I think that this is an important potential future treatment that should be addressed. So we know that epigenetic or chromatin marks are more malleable than genetic DNA sequence changes. There are specific drugs, as I mentioned, that target chromatin are already available and could potentially be repurposed for this type of approach. And we know that this type of approach has worked to improve learning and memory in mouse models of a related disorder called Kabuki syndrome 1. And then my lab is using a similar approach to treat generalized overgrowth in a distinct condition within this group called Weaver syndrome. And I'm just going to show you a little bit of data from that work to kind of illustrate the concepts. So Weaver syndrome is a deficiency of the EZH2 writer of this H3 lysine 27 trimethylation silencing mark. So this is a different mark than we've been talking about all day with KAT6. But the point is that it's also a writer of a particular mark. And so if you have a deficiency of this writer and you can't place this silencing mark, then our hypothesis was that this could alter the chromatin state, causing there to be less of the mark, more open chromatin, and then that could lead to altered gene expression. So just to give you a little primer on this condition, it's a bit different than KAT6A and 6B, but individuals with Weaver syndrome exhibit striking somatic overgrowth. So this is a growth chart. So the average height of individuals in the UK is shown in this thick black line. And these triangular points here are individuals with Weaver syndrome. So you can see that they're quite tall. And in fact, their standard deviations can be up to 7 standard deviations above the mean for height. So this girl who's 12 and a half years old is about 6 feet tall. And this is a patient that we diagnosed with Weaver syndrome, and at age 12, she wore a men's size 13 shoe and was quite tall as well. These individuals also have intellectual disability, which is often mild to moderate, and then they can have distinct craniofacial features like a tall, broad forehead, widely spaced eyes, kind of fuller cheeks, and this kind of stuck-on appearing chin. So we generated a mouse model with the most common Weaver syndrome missense variant, the arginine 684 to cysteine variant, which is in the catalytic set domain. And that's what the domain that's responsible for placing this particular epigenetic mark. And when we looked in mouse embryonic fibroblast cells, as the mutation burden increased, meaning as if you compare it to wild type, if you had this mutation on one copy of the EZH2 gene versus both copies, you could see this dose-dependent decrease in the epigenetic mark, and that's graphed here. But the actual protein remained stable. And so this suggested that, in fact, this mutation does lead to a decrease in this epigenetic mark. And so we looked at the mice, and they did have a skeletal overgrowth phenotype similar to the patients. So they had in this particular graph is just body weight, and only the females had increases in body weight compared the Weaver mice compared to the control mice. And throughout, I'll be showing the mutants, the Weaver syndrome mice in red and the wild types in blue. But when we looked at the skeletal, the actual bones, in this case, this is the thigh bone or the femur, we could see that the bones were actually much larger in the Weaver syndrome mice compared to the controls. And this is quantified here. So we were interested then at getting at the cellular level and looking at the relevant cell type that would lead to this overgrowth. And so we were able to isolate cells that would eventually become the cells that would lay down this bone called osteoblasts. And so we isolated the precursors from the mice and grew those in cell culture. And what we found was that in the Weaver syndrome cells, these osteoblasts that lay down bone, they laid down more bone in the Weaver syndrome mice compared to the controls. And you can see that quantified here. And so this seemed to recapitulate what we were seeing in the mice. We next wanted to see if the gene expression was also different in the Weaver syndrome cells compared to the controls. And so we looked at these osteoblasts and compared the gene expression. And this is a volcano plot, which basically shows gene expression on the x-axis and statistical significance on the y-axis. Anything above the red dotted line is statistically significant. And you can see kind of this V in the data. Anything on this side here is upregulated, or the genes are overactive, and anything on this side is downregulated, or the genes have reduced or are underactivated. And so what we can see is, and then the colors just look at particular pathways. So osteogenesis, or the building of bone, genes that promote osteogenesis are highlighted here. And you can see that these are upregulated in the Weaver syndrome cells. And if we look at another pathway that's involved in bone growth, the BMP pathway, we see that there are genes that promote bone growth are upregulated, and those that inhibit bone growth are downregulated. And so it suggested that there was truly a difference in gene expression in the Weaver cells that build bone compared to the controls. So back to this model of treatment. So we hypothesized that if the Weaver syndrome results from a deficient writer of this particular epigenetic mark, then there might be too much of the eraser activity. And so we tried a strategy where we took a drug that would inhibit the opposing eraser. So this is the eraser of the same mark that EZH2 places. It's called GSKJ4. And so we treated the cells in culture with this drug. And what we found, so this is the drug and this is the control where they're just treated with the vehicle. And what we saw was that, again, in the Weaver mice, just vehicle treatment, there was still an increase in laying down too much bone. But when we treated with the drug, we could actually, when we treated the Weaver syndrome cells with the drug, we could actually correct this back towards the normal state. And then that there was actually no difference between the treated cell's ability to lay down bone and the control cell and the untreated cell, wild type cells. And so this suggested that this strategy for treating this particular phenotype or this particular feature might work. So the next question was, what does it do to gene expression, right? Because that's a big question. If we know that we can actually reverse the cellular phenotype or kind of revert it back to laying down the appropriate amount of bone instead of too much bone, but what does it actually do to the gene expression in the cells? And so we looked at gene expression using RNA-seq. And what we found was that when we compared the Weaver syndrome cells to the control cells, so just looking at the effect of the mutation, we saw that many genes involved in osteogenic differentiation and this BMP pathway that promotes bone growth were upregulated. So in the Weaver cells, they were upregulated compared to controls. And when we looked at the effect of the treatment with the drug, we found that these same genes that were upregulated in the Weaver syndrome samples were then downregulated when we treated with the drug. And if you put all of this sort of on the same plot, looking here on the x-axis being the effect of the mutation, and then on the y-axis, this being the effect of the drug, what we found was that most of the genes which fall into either this quadrant here or this quadrant here. And what that means is that if you have a mutation, or if you have the Weaver syndrome mutation, and you have downregulation of particular genes resulting from the mutation, upregulation of the genes resulting from the mutation, then you have downregulation upon treatment with the drug, and vice versa. If you have downregulation in Weaver syndrome, then those genes become upregulated with the drug. And we were surprised to find that it was really about 97% of the shared genes actually reverse the fold change directionality. And so we weren't really expecting that many of them to actually change, but that's what happened. And so now the next step is to try this drug in our mouse model in vivo in treating the live mice to see if we are able to see similar results there. So, in summary, I just want to reemphasize that KAT6A and 6B are really within this broader group of Mendelian disorders of the epigenetic machinery that result from pathogenic variants in epigenetic machinery genes. And that really these disorders, they're genetic disorders, but they have these genome-wide epigenetic consequences. And we care for many individuals with these disorders, including the KAT6 group in our multidisciplinary epigenetics and chromatin clinic, and we'd be happy to see anyone there in our clinic. And then I think it's really important to see that this really kind of points out unique therapeutic opportunities. So there are a lot, there are a lot of other therapeutic opportunities that are out there, but for this group of disorders, there's this sort of unique therapeutic opportunity to potentially target the epigenetic marks which are disrupted and sort of rebalance the chromatin and gene expression as another therapeutic opportunity. So with that, I will just thank the people that did the work in my lab, as well as our collaborators and our funding sources. And again, thank you to the KAT6 Foundation for inviting me to speak, and I'd be happy to take any questions.
So similar to your Weaver syndrome study, do you have, is there a specific target study you could do on KAT6A or B, or do we not have a clear marker like bone growth that makes it amenable to?
Yeah. So that's a good question. I think the first thing is that we don't really understand the full pathogenesis, right? A lot of the talks have said, is it a hypo, the different mutations might cause disease in different ways. So I think that's obviously really important because as some of the work that was shown earlier, you can't have, we know that this is a very dosage sensitive gene, right? So if some of the variants are leading to upregulation or a gain of function, you certainly wouldn't want to treat those the same way that you would treat variants that are a loss of function, right? Because you would have too much or too little of this particular mark. There are certainly epigenetic therapies. If there's a subset of people where there's a loss of function, then potentially you could use a similar strategy using like an HDAC inhibitor, which is the histone deacetylase inhibitor that sort of opposes the acetylation mark. But again, that would so theoretically there are some potential things, but I think obviously we have to know a little bit more about what the pathogenesis is and how people with different variants, what the molecular mechanisms are there.
Thank you so much. I'm also super something about identifying synergies between these epigenetic disorders. And I wonder if you have the plans to study the regulations of the genomic regions of HCK37 methylation. That's the critical factor to everyone here. That's a repressive mark, yeah. And we also assess it in cognitive syndrome and in that cell. And we see a very specific pattern in those. So I see an opportunity to do hack for that.
Yes.
Before, I don't know if it's something that and whether we could do it.
Yeah, that's a great question. So yes, we have plans to do. We were looking at cut and tag, cut and run versus ChIP-seq, but yes, we have plans to do that and to look at that in the osteoblasts. So and then also we are now wanting to look at certainly because Weaver syndrome also has intellectual disability and other neurobehavioral findings, trying to phenotype the mice and identify a cell type of interest to do some of those types of studies in the brain as well. So yeah, I think it would be really interesting to do that for sure.
To determine whether it's an increase or decrease in order to be able to treat, and how do you do that, and what do you see in patients being treated, what have been the benefits you've seen or the changes you've seen when you go through that?
Yeah. So we have not treated any patients. Only we're only working on this in mice for now. And right now we've done the cells in culture. But the hope would be that it would be able to kind of target the underlying change, or target the epigenetic marks in different tissues, and potentially treat hopefully more of the symptoms rather than having to treat each one individually. And I think as to your question about what do we need to do to determine the mechanism, I think everybody, all the people here, all the scientists here that have spoken today are all, trying to work on understanding the mechanisms, and what's actually going on at the level of KAT6A and KAT6B, and the different variants and the different parts of the gene and the different types of variants. And so I think all of that work is ongoing already. It's just a matter of trying to figure it out, and know kind of what is happening with the different groups, and then potentially just identifying a particular drug that kind of would cause the opposite to happen. Like teenager or adult or whatever use. That's a good question. So we haven't, done that in our studies yet, but there are some other studies in the literature. So our colleague Hans Bjornson, who works on a related condition called Kabuki syndrome, has used this type of approach in a couple of different papers, just a little bit different drug versus a dietary approach, and has shown that in mice, if you treat young adult mice with one of the three ways that he's done this, and sort of with the same idea of kind of tipping that balance of chromatin in the other direction, that if you treat young adult mice with these drugs, that he was able to show that they did reverse the learning and memory defects. So suggesting that postnatally, that is something that could be done. However, again, it could be different for different disorders. So it doesn't mean that that would necessarily be the case in Weaver syndrome or in KAT6A, KAT6B, but that's what was shown in Kabuki syndrome. So I think, again, emphasizing just all the really great work that's going on in the mouse models and the human models and the fly models to get at some of these questions. To see if this might be something that would also work in these conditions. One more question. And human conditions and some other disorders, right? I get questions from all of his grandparents, generally the next day. Oh, could this help? So, Dr. Fung, could you speak in general about all the technological advances with gene therapy for other disorders that's happening, and how those are or are not specifically relevant to KAT6, A, and B, and what the future may hold? Yeah. Okay. So I think, certainly gene therapy, and then there's sort of CRISPR gene editing, and then those can be sort of combined. So I think it's very exciting. And I think you mentioned earlier, like a paper that had come out recently, like in the New England Journal of Medicine, about another rare disorder where they were able to deliver using gene therapy to CRISPR, basically to edit the copy of the gene that had the variants, and kind of deliver that to a child into the liver, and it was shown to correct that. And in short-term studies, it seems like it was a really great treatment. And so certainly these are out there. I think there are, and it's exciting for all genetic conditions, but a lot of these gene therapies tend to work well in when you can deliver the gene, the part in the gene therapy, when you can deliver it to a particular site in the body. So for example, to the eye, like there are some eye disorders where you can deliver gene therapy directly into the eye, and it can work really well and correct disorders. This other example from the New England Journal, they delivered this gene therapy vector that was able to edit the enzyme for this rare genetic condition directly essentially delivered it to the liver of this child, and that seemed to correct that finding. I think it's a little more challenging in this group of disorders because they do tend to affect sort of many parts of the body, and it is inherently harder to deliver things into the brain than to some of these more localized sites. So I think that's still something that's going to cause that's going to be a little more challenging in these disorders. And so that's why I think while these are still very exciting possibilities, I think that continuing to look for other ways that we might be able to treat these conditions is a good idea as well. Sure.
Interesting research.